All right, so we started talking about this animal PowerPoint a long time ago. Going to continue along. In general, these characteristics that you see up here on the screen are the characteristics of animal cells. All animal cells are eukaryotic. They have a membrane-bound nucleus and membrane-enclosed organelles. All of them do. All animals are multicellular. All animals have to eat. They're heterotrophic. No animal cells have walls. All animals, at least at some point in time in their life cycle, are free to move. And most of the time, animal cells are diploid. There's some exceptions, like sperm and eggs. But most of the time, animal cells are diploid. That being said, another thing that we spent quite a bit of time talking about was how metazoans develop as embryos. And remember, that word metazoan means an animal that has what? Tissues. And for purposes of this class, and really the only one I can think of, the only animal that I can think of that's not a metazoan is which one? What's the most simple animal? Sponge. This is a big lump of cells. Yeah. Squid has tissues. Yep. Squid's pretty complicated. We'll get to that in a minute. But the most simple animal is a sponge. It doesn't really have any organized tissue layers. But pretty much all other animals do. So in step one of embryonic development is fertilization. Haploid egg, haploid sperm get together in people where? Fallopian tubes. Those eventually make their way down to the uterus, develop into a zygote. The zygote will go through cleavage. It'll split really fast. So fast that it doesn't really get any bigger, it just makes more cells. So that's step two, cleavage. We'll eventually end up with a solid ball of cells. That's called the morula. And my marker's a little off, as you can see. But the cells in that ball secrete a liquid that ends up pushing a cavity into the middle. Once it's organized into like a hollow ball, those cells are called the blastula. And eventually, the cells in that blastula are going to move around. And they're going to organize themselves into 3 layers of cells, which we've been calling germ layers. But we know the cells in those germ layers are also known as what? What kind of cells? Stem cells. Cells that have the ability to be all they want to be, right? Make anything that they want to make. They haven't differentiated yet. They haven't decided what they want to be when they grow up. Their fate hasn't been determined. In a metazoan, most of the time those germ layers organize themselves into 3 distinct layers. And once they get in those 3 layers, the ball of cells is called the gastrula. We'll look in a minute about a couple other things about the gastrula. But eventually those germ layers are going to start to make things, make parts, make organs. And that's the last part of embryonic development, organogenesis. All that we've talked about before. Point out one thing in this picture. Here's a picture of a gastrula. And you can see the outer layer of cells, the ectoderm, the inner layer, the endoderm. And you can also see this opening right here into the gastrula, the blastopore. In most kinds of animals, that opening becomes what? The mouth. And what do we call those kinds of animals? Protostome. Proto means first. Stome means mouth. Mouth forms first. Other kinds of animals, like the vertebrates, all the vertebrates. And by the way, what's the name for the vertebrate phylum? Chordata. So, chordates, things with a backbone, that opening becomes what? Anus. And they're called what? Because of that. Deuterostome. What does deutero mean? Second mouth. And it says mouth forms second. Other than chordates, what other group of animals includes deuterostomes? Echinoderms, which are things like what? Starfish and such. And then that opening forms a cavity into the gastrula. that cavity is eventually going to help to form what part of your body later? The GI tract, what we might call the alimentary canal, the gastrointestinal tract, the digestive systems. And when it's in the embryo, it might be called the primitive gut. It also is sometimes called the arc enteron. That's that cavity inside the gastrum. But I want to go a little bit deeper into a couple of these things today. This idea of protostomes and deuterostomes. Protostomes include three main groups of animals. And let's just start here. Annelids are protostomes. What's an annelid? Segmented worms, like earthworms and leeches. What about mollusca? What are those things? Yeah, those are things like snails and slugs, oysters and clams. What else? Mollusks. Yeah, they're mollusks. Squids, octopi, those kinds of things. Octopi. And arthropoda, literally, what does that word mean? Arthropoda. Not quite. Pride means foot. What's arthro mean?
Many.
Like arthritis. Arthroscopic surgery. It means joint. Jointed foot. Nice job. Jointed foot. These are like insects. But not just insects. Also things like lobsters and crabs. What do we call those? Shrimp. Crustaceans. These are your insects and your crustaceans. Also things like little millipedes and centipedes. They fall into this group too. All of those things develop the mouth first. And then the deuterostomes, we said, are two main groups. And again, what are those two main groups? Two main phyla, chordata and echinoderms, like starfish. Now, if you look, there's a lot of things that are missing. There's a lot of animals that aren't listed in those 5 groups we talked about. What are some animals that just aren't up there?
Bears.
Bears are up there.
Amphibians.
Where would bears be? Chordata. Backbone. I think amphibians have a backbone too. I think they would be in chordata. Pretty sure they do. Things like roundworms, like hookworms and pinworms. Flatworms, like tapeworms and things, they're not up there. Jellyfish, sponges, none of those things are up there. And the main reason they're not up there is why? Most of them don't have the 3 germ layers that we're about to talk about. What happened? No.
Don't sponges have those holes?
They have pores. Now, you need to know a little bit more. You need to know a little bit more than that protostomes, the mouth forms first, and deuterostomes, the anus forms first. You need to know a little bit more than that, because there's more differences. Maybe the first thing you ought to know is that after fertilization, we know what comes next. Fertilization and then cleavage, splitting. They cleave, they separate in a different pattern. Protostomes, they have what's called spiral cleavage. Forms a spiral-shaped structure when they divide. Deuterostomes, like us, have more what's called radial. But maybe more important than knowing those two things is that in protostomes, after their type of cleavage is also said to be determinate. What do you think determined? What is determined by determinate cleavage? Once those cells split off, their future is determined. They're really no longer what? Stem cells. Their fate is determined. They're going to be something, and they've lost the ability to be other things. See what I'm saying there? Once they've divided off in this early stage, their fate is set. They lost that ability to do anything. Whereas in us, even after cleavage for a long time in embryonic development, our cleavage is said to be indeterminate. Which means even though we made a bunch of new cells, all those cells can still what? They can do anything they need to do. Their fate hasn't been determined. They're still what? They're still stem cells. They still have the ability to make any part of the body they need to make. Now, another part of this I want to talk about is what's called the coelom. Now, that's a term we hadn't really mentioned, I don't think, in here, but it's an important term. What is a coelom? It's a body cavity. And this is really one of the reasons that some of those animals were left off that whole protostome, deuterostome dichotomy thing. There are several kinds of animals that just don't have a body cavity. Why would you not need a body cavity? I mean, we need a body cavity. Why do we need a body cavity? We got a lot of organs, and the cavity is a place to put the organs. The jellyfish doesn't really have any organs. A flatworm has very few, if any, organs. So those things that aren't protostomes or aren't deuterostomes, they're not one of those two things, typically because they don't have a coelom. They don't have a body cavity like us. Now, notice what it says here. The body cavities, the coelom, it's made in two different ways. In protostomes, if you look at how it's made, these solid groups of mesoderm, the germ cell layer, those split off to make the body cavity. So it forms in between those two masses of mesoderm. Whereas in us, the vertebrates and the echinoderms, if you notice right here, here's that arc interrun. Here's the blastopore. And then this is that tube that's going into the gastrula. Notice that it sort of folds up back here in the back, in red, in this area. And those folds end up making what later will become this whole body cavity that holds all your organs. Key thing, both of these body cavities, if you see this word coelom, it's made, the coelom is always lined with tissue from the embryonic mesoderm, which is which germ layer? The middle one, the one in the middle. Kind of makes sense. Body cavities in the middle of your body. It ought to be made by things in the middle. And then the other thing that we mentioned, and we've already talked about over and over again, in protostomes, the blastopore, that initial opening where becomes the mouth, and in deuterostomes, that initial opening becomes the anus. So you ought to be able to explain this diagram in the way that we just did.
The coelom is made from
Mesoderm. It's lined with mesoderm. I'll skip that one for right now. This is a slide we've already seen. Not going to take a lot of time to talk about it. But remember that the three germ layers, endo, meso, ecto, they're stem cells that are in charge of making certain parts of your body. The endoderm tends to make the innermost stuff, like the digestive tract, the lungs, the liver, the pancreas. The mesoderm tends to make things in the middle areas of your body, like the lining of your coelom. The dermis, the middle layer of skin, the bones, the cartilage. And the ectoderm tends to make things on the outer parts of your body, like your eyes and your ears and the enamel of your teeth, and the outer layer of your skin, the epidermis. Here we get a little bit more into this coelom stuff. There are some animals that are said to be acoelomate. That means, literally, they have no body cavity. And the only way they can get by without having a no body cavity is if they have no what? No organs. So these would be things like sponges, jellyfish, some of your flatworms, like tapeworms maybe. They're very simple animals with no real organs. Few, if any, organs. Let's put it that way. There's one phylum of animals called nematodes. What's a nematode? Those are roundworms. Now, we're not talking about earthworms. We're talking about usually microscopic worms, like the worms that cause hookworms or guys ever seen a dog with heartworms before?
Yes.
One of the coolest things that I've ever had to demo to students, and I don't know what happened to it. A vet a few years back had been treating a dog that had heartworms, and he had taken the heart out of the dog, and he put it in some kind of preservative, and he gave it to him. And it was dog heart, and it looked like it had spaghetti coming out of it, literally. The aorta, the main blood vessel, was full of this spaghetti-like stuff, and it was coming out. Every blood vessel going into or out of the heart was full of this stuff. There were even holes in the heart that had been bored through, and you could see these worms coming out. How does a dog get heartworms?
Yeah, eating something.
Nope.
No.
Mosquito bites. The mosquitoes transmit the worms from dog to dog. Now, lucky for us, our immune systems don't let them affect us. But you get bit by some of those mosquitoes. Sure. It's just that your immune system doesn't let that bother you. The dogs, they're susceptible to it. But that's an example of a roundworm.
So we can't get one.
We don't get heartworms. I know it affects dogs. I'm not sure about cats. Anybody know if it affects cats? How what?
Why did you lose a heart?
He may have got it back. I didn't lose it. I just don't remember what happened to it. He probably wanted it back. I kept it for maybe a year or two. I bet he wanted it back to put in his office. But it was really cool to look at. It's gross, but cool to look at. Oh, where I was going with nematodes is they are a little bit more complicated than things like jellyfish and sponges. They have some organs. They have to have a space for those organs. But they have what's called a pseudocoelom. What's pseudo mean? It's false. It's not false because it's not a cavity. It is a cavity. But it's made in a different manner than the coeloms of most animals. Instead of being made from the mesoderm, it's made in a different way. It's made between two layers instead of being made of just mesoderm. So really all you need to know about a pseudocoelom, it is a body cavity. It's the kind of body cavity that roundworms like nematodes have. But it's made in a different way than the body cavities that most other animals have. It's made between the endoderm germ layer and the mesoderm germ layer. So it's like a pocket in between those two.
And then a normal one is made between.
It's lined completely with tissues that came from there, the mesoderm. But if you see the word coelom, realize that's the type of body cavity found in most animals. Annelids. And again, we said annelids are what? Segmented worms, like earthworms. Mollusks, which are things like snails, and slugs, and clams, and oysters, and squid. Arthropods, which are insects and crustaceans. Echinoderms. We said those are like what? Starfish, sea urchins, sand dollars. And then chordates are everything with a backbone. Vertebrates, all the vertebrates. Bears, lions, tigers, oh my, all those things. Yeah, I know. Got a little out of order there, but that's okay. Another thing that we need to know before we get too deep into some of this animal stuff is just some definitions of some terms. A minute ago we talked about metazoans. Sometimes instead we'll say eumetazoans. What's the EU in front? True. So these are animals that have tissues, true tissues. Groups of cells that work together as a unit. Tissues. And as far as I know, I can't really think of any animals other than sponges that don't fall into this category. So parazoans are animals without true tissues. These would be like the sponges. What's para mean? Yeah, close to, next to, almost, sort of like. So that word literally means sort of like an animal, right? Parazoan, kind of like, close to being an animal.
Kind of like.
They are animals.
No, I'm saying.
I got you.
Well, I mean.
It's plant-like plankton. Yeah. Come with me? Anyway, sponges are in a group called porifera, and that's the one group of animals that we're really going to talk about a little bit today. But there's some more terms that we need to make sure we know. Most animals have what's called cephalization. That root, C-E-P-H-A-L. Where else have you heard that root in science or medicine? Cephalopods? Cephalopods, where else? I know what you're going with. What other word is it part of? Syphilis? No, it's spelled in a very different way. It does sound the same. Encephalitis. Heard that word before? Encephalitis?
That's what my cat has.
What is it? You know what it is?
I really don't know what
It's when your head swells.
it is. Yeah, she had a bunch
Swelling of your brain, essentially.
No, she didn't.
What's a cephalopod? No. A cephalopod's like an octopus or a squid. It literally means head and head what? What do we say pod means? Head foot. If you look at a squid, it's sort of got like a big head and then right, okay, head foot. But cephalo means head. What do you think cephalization might mean? It means in most animals, what's found in your head usually other than your brain? Yes. Most of your sensory organs are concentrated in your head. That's what cephalization means. Most of the sense organs concentrated in the head. Now most animals, four-legged. Head goes first. Kind of makes sense that your sense organs ought to be in your head. That goes first, right? You see what's coming, you smell what's coming, you hear what's coming, you taste what's in front of you. So it makes sense that animals evolved to be that way because that's where they're going to run into problems oftentimes. It's when they're moving forward. You need to see what's coming, hear what's coming, smell what's coming. But again, cephalization is when sense organs tend to be concentrated in the head. Another thing that we need to know is just a few directional terms. If we're talking about a four-legged animal, where's dorsal? Yeah, it's like the top where the backbone is at, right? Like the dorsal fin in a shark. It's along his backbone. It would be the top of the dog's body. Ventral is the belly side in a four-legged animal. Anterior means what? Anterior means the front, which in a four-legged animal would be that head side, right? And posterior would be the tail side, the back side. Not the back as in backbone, but the back end, tail side. So make sure you know those terms. Dorsal, the top of a four-legged animal along the backbone. Ventral, the belly side. Anterior, the head side. And posterior, the tail side. Like I said, the one main group of animals, the one main phylum of animals we're going to talk about today is this one, porifera. A pore is what? An opening, a hole. Sponges are animals, even though they look a lot, maybe more like plants than they do animals. They're animals. And the reason they're animals is because they are eukaryotic, multicellular, no what? No cell walls. They are heterotrophic. They can't do photosynthesis. And at some point in time in their lives, they can what? They can move. Not as adults usually, but as larvae, sponges have the ability to move around. They are considered typically to be the most simple animals. They have no true tissues or organs, which means they're just big hunks of what? Fairly unrelated cells. Cells that aren't working very well in groups. They don't have a circulatory system, which means how do things move inside their body? What's the only thing they've got to move things around? If you don't have any circulation, how are things going to move?
They just move in and out. They have pores. They can like go in.
Yeah, but how's it get in though? Diffusion and osmosis. That's what they got, pretty much. Diffusion and osmosis. There's no blood. There's no heart. There's diffusion and osmosis. Is that very efficient? Probably not. Are they going to be very thick? Can their bodies be thick? They can't be, because diffusion, what do we see in the, those little chunks of gelatin, when the gelatin cubes got thick? It became very hard for what to happen? For stuff to get to the middle. They can't get very thick. They're going to have to be very fairly thin, fairly fragile. They're acoelomate, which we just learned means what? No body cavity. They don't need it. They don't have any organs. Now this word we haven't really used before, but you probably can figure out what it means. They're not triploblastic. That means their blastula does not have 3 germ layers. They don't have 3 germ layers. So they have no gastrula stage. They don't have the 3 germ layers that we have. Now in terms of what they eat, they're filter feeders. What are they filtering out of the water? Probably little plankton, pieces of algae, protists, maybe bacteria, small, mostly microscopic things. They're filtering out of the water and feeding on that. Algae. Yeah. A lot of them are hermaphrodites. And that means what? One organism, both sexes. And as adults, they're pretty much all sessile. What does that word mean? Can't move on their own. And for those last few things, I want to go to a picture and explain those last couple of things. Here's a general picture of a sponge. They've got a big hole in the top. You see that right there? That hole in the top is called the osculum. And then inside, they're basically hollow. Along that hollow cavity, they've got these cells with flagella. And they're called collar cells. That's written up here somewhere, I think. Anyway, collar cells. Yeah, it's down there near the bottom. And those flagella whip around and do what? They pull in water. And as water comes in, those are the filters. Those cells filter out the bacteria, the algae, the protists. And then once they finish filtering it, the water comes in through these pores into this cavity. And then once it's filtered, it goes out the top. So it's sort of a one-way flow. In through the pores, out through the top. In through the pores, out through the top. And these collar cells in here are in charge of filtering it out. Get anything that the sponge can use out of the water. And then just sending it on. So there's not much to them. Pretty simple. That's about as complicated as it gets. Now, do they have bones? No. Here's something I bet you don't know. You probably have in your kitchen at home these red, blue, green, yellow sponges that you can take and you can bend around and you can twist up and you can wash dishes with. Are those really sponges? What are they? Those things that we call sponge, that we wash our dishes with, or use to clean out the bathtub, or wash the car. Guess what they actually are? They're made of oil. They are man-made products from petroleum. A real sponge, you might find at places like Bed Bath & Beyond or somewhere like that. You've seen real sponges in there before? Kind of stiff, kind of hard. It probably wouldn't be all that comfortable to try to wash with it because it's kind of stiff. Might even have little pieces in it that are made out of glass. Let me talk about that. So typically, Their support structure is two or three things. No bones. They either have proteins called spongin, or they have these little things called spicules, which are like little sharp spikes made out of silicon dioxide, which normally, what do we call silicon dioxide? Glass. See little pieces of like glass or even calcium carbonate, which most people around here call what? Limestone. So their quote skeletons are either made out of glass or rock, essentially. Kind of hard. They're not that soft, gooey stuff you think of when you think of like a kitchen sponge. That's not what we're talking about. Those are not living things at all. Those are pieces of petroleum, oil. In terms of how they reproduce, they can reproduce asexually or sexually. One way they reproduce is just by budding. And that simply means they grow a bud, probably growing that bud by what process of cell division? Mitosis. And eventually that bud breaks off and makes a new sponge. It's cloning. Another way they can reproduce though is sexual. We're going to save this genial thing for later. We may come to it later. Typically what will happen is they'll release sperm into the water. And those sperm swim around, try to find eggs in another sponge. What does that mean about the number of sperm they're probably going to have to make? A whole bunch. Because what are the odds? In the ocean, you release some sperm, and they find eggs from another sponge. Not very big. So if you want to keep yourself going, your species going, you better make a lot of sperm. So they do. And once the sperm and egg get together, and you get fertilization, they get a larva. It's flagellated. It's got flagella. It looks like a protist. It might look like an amoeba or maybe even like a paramecium. If it's got flagella, what can it do? Well, it can move. It's motile. What's it going to move around looking for? A nice rock, somewhere to put down roots, literally. So it finds a nice rock, basically glues itself to it, and grows up to be a sponge. It's kind of cool. Yeah, it's kind of cool that they do that. But now, they don't have eyes. They have sensory receptors though, so it's more of a touch kind of thing, than a sight kind of thing. Most are, but I see where you're going. You're saying why don't they just reproduce with themselves?
No, but like how do which one produces the sperm and the eggs are first?
Well, they're all doing it. So when they reproduce, they're probably getting pregnant and making others pregnant. See where I'm going with that? Anybody ever been to a coral reef and been like snorkeling on a coral reef? No. One? Just me and Travis. We're the only ones. Okay? Really cool. If you ever do that, you go down to the Caribbean or Bahamas or wherever you want to go, you're probably going to see some sponges. And most of those sponges are like brightly colored. But I'm going to tell you in reality, sponges are kind of dull. Brown, gray, not colored at all. So where do the colors come from? Yeah, somebody's reading. They have things living on or in them. Algae usually, maybe bacteria, usually algae that either live on or in them in a symbiotic kind of relationship. Let's just assume it's algae, which is normally what it's going to be. What are the algae going to give to the sponge to make this worth the sponge's while to partner up with them? Food. They do photosynthesis. And in return, what's the algae getting? It's getting shelter, protection, a home. It works. What do we call that kind of symbiotic relationship? Good for both. Mutualism. It's good for both. It's a mutual agreement. Good thing for both. So here's some sponges without any color at all. That's probably a normal sponge. And then over here you see some reds and some yellows and kind of oranges. All those colors are probably coming from symbiotic algae living inside the sponge. The last group, I'm going to mention this group, not going to go into it much at all, but Phyllonadaria is probably the second most simple group of animals. And pretty much all I'm going to say about them today is give you some examples. Cnidarians are things like jellyfish, sea anemones, coral, and hydras. What do they all have in common? Well, there's a lot of things they have in common, but what's the first thing that ought to come to your mind when you see them? What does the cnidarian part refer to? They have stinging cells. Stinging.
I didn't know.
Well, they don't necessarily sting us, but that's how they get their food. How many of you have been stung by a jellyfish? Not a good feeling. There are several kinds of jellyfish in the world, like the box jellyfish, that can kill you. Okay? Not just hurt you, but kill you. Most of these things, though, I mean, if a jellyfish really had a real brain, would it want to sting you? No. What's its stinger there all about? It's trying to catch food. It's trying to fish. Essentially, it's what it's doing. It's not smart. It doesn't have a real brain. So when does it shoot its stingers? Whenever something bumps into it. So it's an accident that it's getting you. It's not even thinking about what it's doing. It's a reflex. It doesn't mean to get you.
Yeah. What makes it sting over the organisms? Like neurons and their...
It has a simple nervous system, but not like us. It's more of a reflex arc kind of thing. So when it bumps into something, bam, stingers. Sea anemones are most famous probably from where? Finding Nemo. And Nemo was what kind of fish? Clownfish. And he was immune to what? The stingers from the sea anemones. We'll get more into that next time. Phyllonadaria is where we will pick up next week. There you go.